Maybe we can start now. Hi, everyone. Welcome to join us for this webinar. Today, we'll talk about the autoimmune nephropathy animal model we have generated at GemPharmatech. We'll introduce the pathogenesis of the autoimmune nephropathy and choose three common autoimmune kidney disease models: lupus nephritis, IgA nephropathy, and C3 glomerulopathy to introduce their features and how to choose a suitable model for drug efficacy based on drug mechanism action. Chronic kidney disease is a progressive condition that affects less than 10% of the general population worldwide, amounting to less than 800 million individuals. Chronic kidney disease has emerged as one of the leading causes of mortality worldwide. This figure shows the gender distribution of the renal disease according to pathological finding in central China. From this list, we can find that the autoimmune kidney disease are common renal disease, like the list in the primary chronic glomerular nephritis, the membranous nephropathy, the IgA nephropathy, the membrane proliferative glomerular nephritis, and the G3 glomerulopathy. Those kidney diseases are all autoimmune kidney disease. And in the secondary gene, like the lupus nephritis, the HSPN, the ANCA-associated gene, and also the anti-GBM nephritis, all these kidney diseases are the autoimmune kidney disease. So the autoimmune kidney disease affects lots of persons. The autoimmune kidney disease is the glomerular and vascular damage caused by abnormal immune function. Although the driven mediators for autoimmune renal disease are complicated, common pathogenesis is involved in the disease progress. Usually, they were triggered by a variety of antigens, including the exogenous antigen like cationic bovine serum albumin, which was observed in children with membranous nephropathy. Phospholipase A2 receptor is an in situ antigen expressed by the human podocyte, and it is the most frequently detected autoantigen in primary membrane nephropathy. Refer to the extrarenal antigen. The example is the Gd-IgA, which is thought to originate from cells in muscular tissues and cause IgA nephropathy. Another feature is the formation of immunocomplexes. It could be inside immunocomplexes or circulating immunocomplex deposit in the kidney, which will active complement pathway. The consequence of immunological conflict is renal cell and local immune cell activation, which leads to the glomerular injury. We have compared the pathogenesis of the three most common autoimmune kidney diseases: IgAN, C3 glomerulopathy, and lupus nephritis. The learning of the pathogenesis will help us to understand the animal model generation process and help us to choose the suitable animal model for preclinical drug study. The antigen that triggers IgA nephropathy is the galactose deficient IgA. It was recognized as an autoantigen by autoantibodies to form pathological immunocomplex and active the complement through alternative and lectin pathway. IgA has been identified as a dominant or codominant immunoglobulin in the immunocomplex in the mesenteric area of the kidney. Dysregulation of the complement alternative pathway is fundamental to the manifestation of C3 glomerular acquired or genetic alteration of the regulatory proteins of complement pathway result in C3G. The disease is characterized by low C3 level in serum and C3 dominant staining by immunofluorescence. Lupus nephritis is a common complication in many patients with SLE. Self-nucleic acid is thought to trigger SLE, and when autoantibody attacks the kidney, lupus nephritis occurs. The circulating or intrarenal immunocomplex deposits in mesangial, subendothelium, and subepithelium space. Complement factors are activated by classical pathway or alternative pathway. The treatment of autoimmune nephropathy usually involves the blood control and anti-inflammation. So the medicine affecting the renal angiotensin system and immunosuppressants are widely used as the first-line drugs. Since the autoantibodies production and complement activation significantly contribute to the disease progress, targeted therapies focus on B cells and complement are under intense development. We list the targeted therapies of lupus nephritis and IgAN and also the G3 glomerular based on their pathogenesis in this table. The B cell targeting therapies could be used for the treatment of the three diseases, since immunocomplex are all involved in the disease development. The drugs that targeting alternative pathways like CFB, C5, and C3 inhibitors could be evaluated in the lupus nephritis disease model. The IgA nephropathy animal model could be used for testing lectin or alternative pathway targeted therapy like MASP2, CFB, and C5a receptor inhibitors. The alternative regulators such as CFH, CFB could evaluate in C3 glomerular models. The endothelin A receptor antagonist could also be evaluated in the IgA nephropathy models. This data summarized from preclinical and clinical research also show the targets of three diseases, including the BCL, complement, and IS blocks. So far in GemPharmatech, we have generated a kidney disease model with distinct pathways, like the B6-BAF lupus nephritis model is featured with activation of B cells. The IgA nephropathy disease model is induced by BSA and carbon tetrachloride with activation of actin and alternative pathway. The humanized C3 mouse is the accelerated C3 glomerular model with alternative complement activation. The CFHQ mouse also show alternative complement activation. Our clients could choose the suitable model used in this table to match their drug mechanism of action. Then I would like to introduce the detail of the disease model we have gained at GemPharmatech and sharing the experience for how to use this model for drug efficacy study. First is the lupus nephritis model. The lupus nephritis is a complication of SLE. SLE is caused by autoantibodies that are produced by plasma cell. BAF is the mediator for B cell differentiation to plasma cell and autoantibody and autoreactive B cells. So we generate this model with transgenic the human BAF coding sequence random to the mouse genome and to get this mice model. This mice show display the B cell activation. So this model could be used for study the mechanism of BAF involved disease and evaluation the drugs detecting for BAF and also detecting for B cell. We examined the serum Ig level in this model during the experimental period from 6 weeks to 34 weeks. We observed the increased level of IgM, IgA, and also IgG. And the level of anti-double-stranded DNA in the serum were also increased during the 6 weeks to 34 weeks. And more interesting, these mice display the lupus nephritis manifestation at around 25 weeks. As showing in the pathological study data, the mouse showing the immunocomplex deposition and crescent formation, also when the infiltration of inflammation cells. So all this serum anti-double-stranded DNA level and histological change indicate lupus nephritis is displayed in the B6 humanized BAF mice. We also performed the statistic analysis of the pathological index and the statistic data showing the increased quantitative index of the lupus nephritis in the TG mice compared with the control mice, both in the female group and the male group. And also the chronic index of the lupus nephritis also increased in the disease model, in the TG model, compared with the control mice. We also found the increased staining of C3 immunocomplex in the inflorescence studies. We found the intensive staining of C3 positive in the BAF TG mice, as shown in this data. Then we performed the drug echo study in this model. In this study, we treat the mice with the BAF antibody, the immune-mediated, and after 6 weeks of dosing, we could still find the increased level of IgG, IgM, and IgA in serum. But with treatment of the BAF antibody, it could reduce these Ig levels in the serum. And also the BAF treatment should decrease the anti-double-stranded DNA level in the serum. More interesting, after 20 weeks of dosing the drug, we could find the decreased level of UCA. UCA is the ratio of albumin to creatinine. It's the indicator of kidney failure. So the treatment of the BAF antibody could rescue the kidney disease. And in this pathological study, we also observed the increased mesangial matrix proliferation and basement membrane thickening in the disease model in the BAF transgenic mice. And after treatment with the BAF antibody, the pathological change milder compared with the disease model. In the immunostaining study, we also found the intensive staining of IgA, the IgA showing the increased level in the humanized BAF mice model, and the treatment with the antibody, the BAF antibody, it could reduce the IgA positive staining. Then we would like to know the mechanism of the drug. We performed the flow cytometry study to analyze the subpopulation of immunocell in blood. As expected, the ratio and count of TFH cell, the total B cell, plasma cell, and also membrane cells are all increased in the disease humanized mouse model. And with the treatment of the BAFF antibody, it could reduce this immunocell subpopulation in blood. Similar results was also observed in spleen. The ratio of counts of TSH, total B cell, plasma cell, and membrane B cell are all increased in the BAFF humanized mouse model, and the treatment of the BAFF antibody could reduce the immunocell subpopulation. So the next model is a BSA-induced IgA model. And before we introduce this disease model, I'd like to take 7 minutes to talk about the four-head hypothesis for the generation of IgA. This will help us to understand how to generate this disease model. After pathogen infection in respiratory and in intestinal tracts, plaque cell in muscle tissue secretes the IgA. Increased amount of circulatory Gd-IgA are recognized as an autoantigen by autoantibodies, and they will form the pathological immunocomplex, some of which deposits in the kidney and induce kidney injury. So for generation the IgA nephropathy model, BSA was already administered to mice to trigger muscle immunity, and carbon tetrachloride was used to destroy the liver to reduce the circulating immunocomplex. It will take around 8 weeks to induce a model, and then the test drugs could be evaluated for 4 weeks. In this model, we using 3 conditions to induce this model. And in this group, we found the decreased level of the creatinine in the urine, and also we observed the increased level of albumin in urine. The ratio of UTP to creatinine increased in the treatment group, and also the ratio of albumin to creatinine increased in the treatment group. We also observed the increased level of IgA in the serum in the disease group. And after 8 weeks of treatment, we also observed the increased level of albumin in urine, and also increased level of the ratio of albumin to creatinine. The IgA level in serum will also increase after 8 weeks of dosing. And we observed the positive staining of IgA in the BSA treatment group, and also the IgG positive staining were observed in the BSA treatment group. The HEE data also showing the infiltration of inflammatory cells in BSA treatment disease group, and the BSA disease group also showing the manifestation of glomerular nephropathy. Kidney path staining showing the hyperplasia of glomerular mesangial. Then we performed the drug efficacy study using this model. In this study, we used the CSB inhibitors and angiotensin A receptor antagonists to treat this model. First, we established the BSA-induced disease model, and after 8 weeks of treatment with BSA, we observed the increased IgE level in serum, and also the ratio of UTP to creatinine, as well as the albumin to creatinine. And with the treatment of the drugs, 2 weeks of the dosing, we could see the decreased level of UTP to creatinine in the drug treatment group, and also after 4 weeks of dosing, we found the ETA inhibitors could significantly decrease the level of IgA in serum, and also the CSB inhibitors, the ETA inhibitors, all decrease the UTP to creatinine ratio significantly compared with this group. The disease model group also displays the renal anemia, and the drug treatment could alleviate the manifestation. So this BSA-induced IgA model could be very helpful for evaluating the drugs detecting for IgA, especially the CSB inhibitors and the ETA inhibitors. The next model is the B6 CFHQ mice. Dysregulation of the alternative pathway can lead to C3 glomerular. CFH is a negative regulator that interrupts the C3 convertase. The CFH is a negative regulator that will interrupt the C3 convertase forming and blocks the activation of complement. Dysregulation of CFH has been observed in C3 gene patients with decreased C3 level in circulation. So we generate this model with knockout of CFH in B6 mice. In this model, we observed the decreased C3 plasma level, which is very similar to the manifestations that have been observed in C3G patients. But it seems there is no impaired kidney function were observed in B6 CFHQ mice, since we do not observe the change of the UCA and albumin in urine even at 34 weeks. And then when administration the mice with the anti-GBM antibody, after 3 days treatment with the GBM antibody, we found the increase of albumin level in the urine in the homo knockout mice. And also we found the increased level of UCA in the homo knockout mice compared to the wild type mice. And the 7 days after treatment with GBM antibodies, we found the significant increased level of albumin in urine in the homo knockout mice compared with the wild type mice, and also significant increased UCA level in the homo knockout mice compared with the wild type mice. So this CFHQ mice with induction with the anti-GBM antibody could be used as the C3G disease model. The last model is a humanized C3 model. We generate this humanized model with random insert human C3 gene into mouse genome, and the human C3 could be detected in TG mice. Due to the lack of crosstalk between the human C3 protein and mouse complement regulator factors like CF, this will leading the overactivation of complement pathway and cause the C3 consumption in this model. So in this mice model, we could observe the overactivation of complement pathway. And in this model, at around 6 to 8 weeks, we observed the increased level of albumin in urine, and also we observed the decreased level of creatinine in urine. The indicator, the parameters that could indicate kidney failure, the UCA also increased at around 6 to 8 weeks. This phenotype were observed in male mice. There is no difference of UCA in female 6 humanized C3 mice. And the pathological study also showing the also showing the inflammatory cell infiltration in the kidney in the humanized C3 mice. We also observed the spontaneous mortality in B6 humanized mice, and the mortality rate of male mice was higher than that of the female mice. As shown by this data, these humanized C3 mice could be very helpful to evaluate the drugs detecting for human C3. So here, we summarize the animal model here again. We have generated the different autoimmune kidney disease model, like the humanized BAF lupus nephritis model, the BSA-induced IgA model, the humanized C3 mice model, and also the CFHQ mice. Each disease model has a distinguished pathogenesis. Like the B6 humanized BAF model, it featured with B cell activation, and it activates the complement pathway through classical and alternative pathway. The BSA-induced IgA model activates the complement pathway through lectin and alternative pathway. So this model could be used to evaluate the drugs detecting for the lectin and alternative, like we can evaluate the drugs detecting for MSP2, CFB in this model. The humanized C3 mouse model is the accelerated C3 glomerular process animal model. This model, the pathogenesis of this model is through activation of complement pathway in the alternative pathway. So in this model, we can evaluate the alternative pathway drugs, like the drugs detecting for the CFB and also detecting for C3. This CFHQ mice is another C3 glomerular process animal model. It could be display the manifestation by induced when the GBM antibodies. So our clients could use this table to choose a suitable model for their drug application. Well, here is also disease model we would like to share with our audience.